For years, the field of genomics has been locked into a rigid equation. To confidently map a human genome, the industry established a strict benchmark that struggled to differentiate biological variations from machine errors, forcing them to overwhelm noise with redundant data. Institutions absorbed this massive financial overhead, treating the need for 30 layers of data as an immutable physical law of accuracy. Deep learning models bypass this linear constraint. This graph compares total errors versus sequencing coverage. The orange line represents traditional methods like GATK4, while the blue maps the deep learning model DeepVariant. DeepVariant achieves the same accuracy at 22x coverage that older methods required 30x coverage to reach. Dropping the data requirement from 30 to 22 translates directly to a greater than 25% reduction in sequencing and reagent costs. With a quarter of the budget freed up, research facilities can sequence entirely new populations without increasing their capital expenditure. DeepVariant changes how the computer processes DNA. Instead of calculating statistical probabilities for each base pair, it frames variant calling as an image classification problem. Aligned genomic read data is transformed into a multi-channel tensor, or pilot image. Each grayscale panel encodes a distinct feature. One channel represents the read base, another maps base quality, and a third tracks mapping quality. A convolutional neural network processes these multi-channel tensors, exactly like facial recognition software processes a photograph. The network is trained on vast data sets to visually distinguish the physical signatures of true biological variants from the texture of systematic machine errors. Because the model learns to ignore artifact noise through spatial patterns, it no longer needs redundant layers of data to force a correct answer. Because it relies on learning rather than rigid statistics, the software architecture evolves without requiring updates to the physical sequencing machines. Certain regions of the genome create highly ambiguous alignments, particularly insertions and deletions, or IND errors. By adding a new information channel, specifically haplotype context, the model gains the data required to resolve complex ambiguous sites. This single addition drives a nearly 40% reduction in IND errors compared to the baseline. By incorporating new biological contexts as training data, the learning engine constantly refines its accuracy and drives down the required coverage depth. This contextual approach scales effectively when analyzing multiple related genomes. A model called Deep Trio applies this framework to family structures, jointly analyzing child, mother, and father genomes as a single computational task. Supplying parental context allows the network to learn Mendelian inheritance patterns, further resolving genomic ambiguities in the child's data. This graph plots F1 accuracy scores across coverage depths for a child sample. Dotted lines represent single-sample analysis, while solid lines map the joint trio analysis. The trio model reaches the same accuracy at 20x coverage that a single-sample analysis achieves at 30x. Adding algorithmic context acts as a force multiplier for accuracy, lowering the budget requirements for large-scale, family-based population studies. The implications extend from research budgets directly into acute clinical care. For critically ill newborns in the NICU, a rapid genetic diagnosis can guide life-saving interventions. But traditional analysis pipelines take days or even weeks. A study led by Stanford University deployed a highly optimized DeepVariant pipeline to accelerate this timeline. This timeline tracks the process from the moment a 2 milliliter blood sample is drawn through DNA extraction and into sequencing. At exactly 7 hours and 18 minutes, the pipeline completed its variant calling and identified a likely disease-causing mutation in the TNNT2 gene. This rapid identification directly informed the patient's care plan, eliminating the need for an invasive cardiac biopsy. When the computational bottleneck is removed, algorithmic efficiency translates directly into clinical velocity. This processing speed requires a specific back-end architecture. The workflow relies on massive cloud parallelism, distributing the compute-intensive base calling and alignment tasks across dozens of concurrent GPU and CPU instances. This panel details the hardware acceleration, specifically leveraging modern CPU instruction sets like Intel AVX 512 to process the convolutional neural network. That specific hardware integration cuts the core variant calling runtime from over 11 hours down to just 3.4 hours. These three dials illustrate the convergence of this technology, achieving high cost savings, cutting time to diagnosis to under 8 hours, and maintaining maximum accuracy. The historical constraint that forced researchers to choose between speed, budget, or quality has been broken. Deep learning has rewritten the economics of sequencing.